Hello everyone, I hope you are doing well. So today we are going to discuss about the further detail of mitochondria. First of all, mitochondria is the site of aerobic respiration as we previously discussed, as well as its inner mitochondrial membrane contains several electron carriers such as cytochrome, NAD, FAD, NAD. First let us discuss NAD. NAD is nicotinamide adenosine dinucleotide. It is, as we previously discussed in our video of nucleotides, that it is having niacin vitamin or vitamin B3, and it is having two nucleotides which are attached to each other. One is one contains nicotinamide, second contains the adenine forming nicotinamide adenosine dinucleotide. And similarly, there's FAD flavonoamide adenosine dinucleotide, in which flavonoamide nucleotide joins with adenosine nucleotide, due to which both of these combine together by phosphatidylester bond, due to which they collectively form the dinucleotide known as flavonoamide adenosine dinucleotide, and it is having flavin, which is derived from the riboflavin, which is made up from the riboflavin, which is vitamin B2. Apart from which, we are having um, a few different structures such as the DNA. Now, DNA of mitochondria is guanine cytosine rich DNA, which is an extranuclear DNA because it is not present in nucleus. It is extranuclear DNA. And apart from this, it is circular, double-stranded, as well as it is having its another bubble. It is also known as empty DNA or mid DNA. These are some characteristics. Apart from which, inner membrane, outer membrane, it is having basically a few particles inside it, which are known as Fernandez Moran particle. As we previously discussed, Fernandez Moran particles are present in the mitochondria on the inner mitochondrial membrane, F naught, F1 particles, in which there are two parts, F naught particle, F1 particles. Now these Fernandez Moran particles, F1 part is having an enzyme which is known as ATP synthetase enzyme, ATP synthetase enzyme. So we are going to further discuss why is it important. So basically, when during Krebs cycle, as we know that NAD and FAD are converted by reduction into NADH and FADH. Here, this NADH and FADH are then further oxidized into the electron transport chain. These are further oxidized into the electron transport chain into NAD and FAD, releasing two hydrogen ions and two electrons. And these electrons are then carried by the electron carriers. These electron carriers, as we previously discussed, are in actual the proteins which are made in the ribosomes of mitochondria, which are utilized in the redox reaction during electron transport chain in the inner mitochondrial membrane. So these electron carriers are in actual the proteins, after which when these electron carriers carry these electrons, and when during electron transport chain, NAD and FAD are oxidized during electron transport chain. When NADH and FADH are oxidized to release electrons, the presence of the electrons causes to trigger or activate the Fernandez Moran particle, due to which the F1 particles utilizes or causes the ATP synthetase enzyme to become activated, due to which the ADP plus inorganic phosphate group present in a ADP plus inorganic phosphate group inside the F1 particles is converted into ATP, which is highly energized bond containing particle. This is ATP, this is inorganic phosphate, both combined together by the energy which was released during the oxidation of NADH and FADH, due to which it forms highly energized bond containing molecule known as ATP. Highly energized bond containing molecule known as ATP. Highly energized bond containing molecule ATP. So during this process, 80% enzyme is utilized in that process. Apart from which, when it comes to a few more things, that here the mitochondria is able to divide itself by its own. It is known as a semi-autonomous organelle. Why? Because it is having its own DNA, RNA, ribosomes, proteins, which are utilized for the formation of electron carriers, enzymes, and utilized by its own cell, not by the not it is utilized by its own mitochondria. It is not utilized for the cell. Secondly, we are having the proteins, after which there is the presence of enzymes, and all of these cause mitochondria to self-replicate by itself, which means it is self-replicative. It's self-replicative, which means it can divide itself by its own through binary fission, and it can divide itself by its own by binary fission. Apart from which, these semi-autonomous organelles, these semi-autonomous organelles are known as semi-autonomous organelles. Also, they should be autonomous, but semi-autonomous because these are neither completely dependent on mitochondria, their own body, and they are neither completely dependent on their own self, nor these are completely dependent on the cell. Why? Because even though they are having their own DNA, RNA, protein, enzymes, they are self-replicative, but they are dependent on nucleus and cytoplasm because nucleus contains some specific types of genetic code of DNA of the nucleus, due to which it synthesizes specific proteins which are required by the mitochondria. And for the requirement of those proteins, for the acquirement of those proteins, the mitochondria requires to remain within the cell in order to gain that specific type of protein structure. Secondly, the cytoplasm contains several micromolecules such as glucose, amino acids, fatty acids, which are required by mitochondria in order to oxidize them and produce energy. If mitochondria is not inside the cell, then there will be no such macromolecules, due to which the mitochondria will have no oxidation, no ATP generation, due to which there will be the degradation, there will be the fatal of mitochondria, which means if there is no energy, then of course mitochondria will not be able to function, because if there is no glucose, no lipid, no fatty acids, no amino acids, then it will be having uh, no energy, no oxidation, due to which it will not be able to survive in an extracellular environment. Apart from which, when it comes to a few things left behind, it is basically uniparental inheritance. What does this mean? This means it is only inherited from one parent. It is only inherited from the mother side, from the mother cell. It is only inherited from the mother cell. Egg cell is not inherited from the father cell. Why? There is a particular reason behind it as well. That's here, as you can see on my screen, that this is ovum. These are two walls outside it. Wall number one, corona radiata. Wall number two, zona pellucida. Zona pellucida made up of glycoprotein. And here we are having the sperm cell, this is sperm head. This is mid region. After which here is the tail. So here is sperm head is basically having its nucleus and other types of stuff. Other organelles and nucleus is present over here. Here this middle region is having the mitochondria present inside the middle region. On the other hand side, this ovum is having several mitochondria inside it. Ovum is having mitochondria inside it, and it is basically bigger in size than sperm. As we are having megaspore. Megaspore in plants is also bigger than microspore, because egg is bigger than sperm. Now, here in this structure, the head is basically having a nucleus, while the middle part is having the mitochondria. Now, as we know that during fusion, there are several lysosomes present inside the sperm cells, which are known as acrosomes. These acrosomes release accretion. These acrosomes release accretion enzyme and hyaluronidase enzyme, which degrade the walls. Accretion acts on zona pellucida as well as corona radiata is having the hyaluronidase acted over them, due to which the coronal radiator made up of lipid proteins, carbohydrates, and hyaluronic acid is degraded by the hyaluronidase enzyme produced by the acrosome, due to which both of these balls are degraded. Now, as these balls are degraded, the sperm enters the ovum, but as it enters the ovum, it is only able to fuse the head part of the sperm with the egg. Head part of sperm is only fused with the egg, while the rest of the part, such as the middle piece as well as the tail, is left behind. And tail is also the property which makes the sperm motile, but it is not even fused with the egg cell. Only the head part is fused with the egg cell, due to which it causes to lack the mitochondria to be inherited from the parental side, but it is inherited from the maternal side, from the mother side, due to which even though ovum contains mitochondria, sperm head does not contain mitochondria. When they fuse together, there is uniparental inheritance. Biparental inheritance, the one in which there are um, two genes, ABC and a small ABC. Now, small ABC. Now, in that case, A, capital B and small b I get replaced of two different genes, due to which B comes on the upper hand side, small b comes here, and capital B comes here, due to which it collectively forms a biparental inheritance. But in this case, there is no biparental, there is uniparental inheritance, and biparental inheritance occurs by crossing over, but here there is no such structure. I hope that you like the video today, and hopefully we'll meet again in another one.